glucose in the end. They really only emphasize the light-dependent reactions which make ATP and NADPH. And the Calvin cycle, which doesn't really do anything if you ignore what happens right here. The Calvin cycle just like takes RUBP and does a bunch of stuff with it, and you end up with RUBP again. And then a bunch of stuff happens and you end up with RUBP again. There's just one last little thing to think about, and I don't think it's gonna blow your minds, but it was really helpful to me when I was first learning this, so I'm gonna explain it to you as well. If you look at what's going on here, what you see is the numbers don't quite add up. If we start with 5 carbons and we add 1 carbon from carbon dioxide, what happened to the other 3 carbons? Like we had 5 here, we add 1, now we should have 6. We only have 3. But we can kind of be like, oh, well, maybe there's 2 of these. So we have 2 of these, so that's 6. The reduction phase leads to 2 of these, so that's also 6. And that leads to, this is more reduction here, that leads to our G3P. There's 2 of these, so there's also 6. So far, so good. And then somehow we end up with 5 again. So we went from 5 to 6 to 6 to 6 to 6 to 5. What happened to that extra carbon? Where did it go? Well, maybe that's part of what goes here, but there were 3 carbons that went off here. So when we look at the Calvin cycle, one of the things that many times people expect you to know and be able to do, and I'm going to expect that you can do this, there's this thing that we call counting carbons. And essentially what it has to do with is balancing all the reactions here so that we have a consistent number of carbons everywhere. And here's what it looks like. If we think about this in terms of the number of carbon dioxide atoms or molecules that it would take to make 1 glucose molecule, we need 6 carbons to make 1 glucose. So if we have 6 carbons coming from 6 CO2 molecules, they will combine chemically with 6 RUBP molecules. These are 5 carbon sugars. So we have 5 carbons here times 6 molecules. That's 30 carbons. We have 30 carbons here. We add 6. We're going to end up with PGA accounting for 36 carbons. That means since this is a 3 carbon molecule, there's 12 of them. So we have 3 carbons times 12 molecules. That stands up during the reduction phase. We have 12 3 carbon molecules here, and we have 12 G3Ps. Those are our 3 carbon molecules here. And we use enough of those to make a glucose. So that's 3 GPs. So that's going to be 2 times 3 carbons, which equals 6. Here we have 1 6 carbon glucose. What does that leave? That leaves, well, we had 36. We lost 6 of them. So that leaves 30 over here, which turns out that it's just enough to make 5 RUBPs. So that's where like the balancing comes in. You can look at this in terms of the number of carbons you need to make a glucose, or the way that I learned it, you can do half of all of this stuff. So we have 3 CO2s, we have 3 RUBPs. That means we have 18 3 PGs, we have 18 G3P, and that goes back to 15, 12, I already lost count. 3 RUBPs, 15 carbons, 3 RUBPs. This is my diagram that I made. I made this years ago, but this is the diagram that I practice, and this is what you should be able to do by memory. So you won't have time, obviously, to write this down now, but this is the stuff you'll need to know. So we've got 3 carbons here, 15 carbons here. Rubisco catalyzes the reaction to 3-PGA or 3-PG. Then we have our reduction that leads us to 6 G3Ps, one of which gets shunted off into the cell cytoplasm to make glucose. The other 5 get morphed around until we regenerate our UBP. That's the piece that you'll need to know for the Calvin cycle. Yeah?
How does the cycle begin if there's
There
no
is-
UBP present?
So there is no real beginning. What you should imagine is that all these chemicals are just present. They're there already. And so at any time, there's gonna be some 3PG that could potentially react with ATP and NADPH and a bunch of enzymes to make G3P. And that's it. It isn't like there's nothing and then it starts. It starts from like already existing. Every chloroplast comes from some other chloroplast. And when those chloroplasts grow and divide, all of this stuff goes along with that. Yeah. Okay. So the big picture of what we looked at today is that we have our light reactions with light as our input, water as our input, oxygen waste as an output, ATP and NADPH as an output. And then in the Calvin cycle, we have CO2 as an input. The ATP and NADPH that we got from the light reactions are used. And this Calvin cycle regenerates one of those molecules over and over and over and over again. But we're adding a carbon from carbon dioxide every time. And eventually, you get enough of those added up to make a 6-carbon sugar, which is glucose, okay? And there's a video that I have posted. You can watch this if it's helpful to you. I'm not gonna play it for everybody, but it's posted in here and it's posted in Canvas. Okay, I'm gonna just show you, I wanna show you the video, the ATP synthase video. It's really short. I wanna show you that briefly, and then I will tell you what the deal is for the rest of class, and you all will have some time to get some work done.
We are looking at the turning gamma subunit and the epsilon subunits, and there's the C ring in the membrane. The F1 complex is outside of the membrane, while the F0 complex is embedded in the membrane.
It's not stuff you need to know. I just want the visual.
And the B subunit. The alpha subunits are shown in light pink. The beta subunits are magenta. In the middle, the central stalk is rotating. The B subunit stabilizes the hexamer, which means that the hexamer can't rotate. We will now look at a rendering of known structural states and the conformational changes that are happening. The beta subunits are seen as a ribbon diagram. The ADP and inorganic phosphate diffuse into the active site, and the ATP has just been released. And simultaneously, the conformational change occurs in the beta subunit. If we zoom into the active site, we can see the molecular structure of the substrates and the chemical reaction. Here is ADP with its 2 phosphates. They are all held in place by a network of hydrogen bonds mediated by the amino acids in the folded structure of the beta subunit. Hydrogen bonds stabilize the base and the sugar, but really primarily the base. There's also a magnesium ion that plays a critical role in not just stabilizing, but also neutralizing the charge of the phosphates. Notice the conformational changes that are happening when the inorganic phosphate comes in. There is the promotion of a planar transition state. Synthesis of a new phosphodiester bond is catalyzed, and then the subunit switches to the open conformation. Upon this reconfiguration in the beta subunit, ATP is ejected from the active site. The enzyme then resets and restarts the synthesis of another ATP molecule. This is a summary of the events at the end.
Okay, so that's, I just wanted you to see that, and the idea here is it's not that you need to know any of that stuff, it's just, it's kind of crazy to think about. What's going on is the movement is being driven by the flow of hydrogen ions out from the lumen of the thylakoid, and then that movement is changing the shape of a bunch of other structures as that central piece turns and turns and turns, and the energy that is imparted to those structures is then used to catalyze the attachment of an inorganic phosphate group to ADP. So it's a lot. You don't have to know all this stuff, but the energy transformations are kind of cool to think about. Okay, so here is your job for the rest of the day today. Number one, I'm guessing at least some of you have not yet finished the photosynthesis POGL. Number two, there's the AT POGL that I asked you to do for homework. Number three, there's homework over the weekend where this doesn't cover photosynthesis. It's units or sections 3.1 to 3.3, a bunch of multiple choice questions on AP Classroom. And you can work on any and all of this stuff. The POGLs are due today, theoretically. The homework is due on Monday, theoretically. But I'm not gonna be overly worried about late penalties. So if you need a little bit of extra time to get these things done, then take the time. And when I score it, I'm just not gonna worry too much about when things come in. If it's more than a couple of days late, then you can expect to lose points. But if you don't get all this stuff done until Monday or Wednesday of next week, that's fine. If you need a break, you can take one. Does anybody have any questions? Yeah, that was brutal, huh? Okay.